Hello and welcome to CSIR NET Answer Key discussion for September 2022. Kindly note this is for life sciences of the morning session and the unit to be discussed here is Unit 1, that is molecules and their interactions. The first question, number 1 for part B. In crystalline NaCl, how many chloride ions surround each sodium ion? They have options 4, 6, 8, 10. Now NaCl is supposed to be an ionic molecule. And the ions, the property of ionic molecule is that the ions will remain bonded close to each other from whichever direction is possible. And they will be present in the form of a lattice or a crystal form. So if you look at NaCl, the sodium is surrounded by total 6 chlorine. That is, if you just check this green molecule over here, here we have 1 chlorine, here we have 1 chlorine. And apart from that also in the sides also we have this chlorides coming out. So that way you will have total 6 chlorine ions surrounding the sodium ion. Thus in sodium chloride the coordination number is 6. Here the correct answer would be option 2. This is from subunit 1A, that is structure of atoms, ions and molecules and also chemical bonds. Next question, question ID 2. At which one of the following electron transport chain complexes does antimycin A inhibit the respiratory chain? We know the respiratory chain consists of complex 1, 2, 3 and 4. This question is from metabolism of carbohydrates, that is electron transport chain. And if you look at, if you know about the inhibitors, it's a very important topic. We have several inhibitors inhibiting lot of complexes. The complex 3 is specifically inhibited by antimycin A. It's also called a cytochrome BC1 complex because in this complex you have cytochrome B and cytochrome C1 molecules, but it is also called as ubiquinone cytochrome oxidoreductase. Now antimycin particularly blocks the electron flow from cytochrome B to C1. And if the electron flow is not there, then the entire respiratory chain will be inhibited. So here the correct option would be option 3, that is complex 3. Next straightforward question from part B, question ID 3. Identify the ribose conformation in the nucleotide shown below. So these are, we know sugars are present in the nucleotide, one of the component along with the base and the phosphate. Now the sugar has some levels of conformational changes because carbon being a tetrahedral atom, it tries to be in a more comfortable position, and that's what is called as sugar puckering. Now if I show you the sugar puckering, the pentose ring in nucleotides is not planar, but it occurs in one of the variety of conformations which is called as puckered. Especially this ribose rings, ribofuranose in nucleotides. Furanose means where 4 carbons are in the ring along with an oxygen. They can exist in 4 different puckered conformations. In all the 4 cases, 4 of the 5 atoms are in a single plane in all these cases. So that means out of these 5 atoms which are part of the ring, 4 are in the ring but 1 is outside the ring. As you can see, this atom is slightly raised out of the plane of the ring. Similarly here, this atom is slightly raised from the plane of the ring. Rest 4 are in the same ring. So this is what is either depending on what atom is out of the ring. For example, here C3 dashed, we know that basis, the one which is connected to basis number 1, that is anomeric carbon. This is number 2, number 3, number 4 and number 5. So we label it as dashed because bases also have carbons which are named as 1, 2, 3, 4. To avoid confusion, we have the numbering of the ribose sugar with dashed. Now here 3 dashed carbon is out of the plane. Okay. So what we can call here is 3 dashed endo. It is facing towards this C5 group. So it is C3 dashed endo conformation. This is typically what we observe in A DNA form. And another one is where 2 dashed form is out of the plane facing the C5 side. So that is called as 2 dashed endo. Now observe that what is 3 dashed endo is also 2 dashed exo because the 2 dashed is out of the plane here. And what is 2 dashed endo is also 3 dashed exo. So this is what we observe 2'-endo in B conformation, or we can also say 3'-exo is also observed in B conformation. Now here they have given clearly, we can see this is the C1 dashed, this is the C2 dashed. So C2 dashed is in line with the C5 dashed. So this is C2 dash endo, and this is also C3 dashed exo. So we have to see what is the options. So definitely not this. So this is also not correct. And so number 1 is the correct option. Option 1 is the right answer. This is from subunit 1B, which talks about the structure of nucleotides. Very specific type of questions they ask from this subunit. Next question, question ID 4. Which one of the following correctly describes the effect of mutation in phosphofructokinase, PFK? PFK is of two types, so PFK1 and PFK2. PFK1 is the enzyme which is involved in glycolysis. It's one of the key enzymes in the third step of glycolysis. That regulation they have asked. Again, a question from metabolism of carbohydrates. So this leads to only to the loss of allosteric regulation by ATP. So you have to say if there is a mutation in this PFK such that it will have only loss of allosteric regulation by ATP, then what will be the effect over here? Okay. So PFK, we know it catalyzes the rate limiting step for glycolysis, and it is allosterically controlled. This is an allosteric enzyme. Allosteric enzyme means apart from substrate binding site, some other site is also there where modulators can come and bind to it. If I show you the PFK, PFK catalyzes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Now, the entire process of glycolysis is ultimately for the production of ATP. So if ATP is already present in the cell, it is a negative regulator that will tell PFK not to synthesize any more product, no more product formation is required. So it's kind of like a feedback. So ATP inhibits this particular enzyme. And on the other hand, AMP and PI positively regulates it along with some other molecules also. So all those small amounts of ATP are essential for the phosphofructokinase reaction because the reaction itself needs ATP. The ATP is a part of the reaction because it has to be coupled with the reaction for it to take place. But at high concentration of ATP binds to a special inhibitory site, that is the allosteric site, and it changes the shape of the substrate concentration curve from hyperbolic to sigmoidal. Because it's regulatory enzyme, that's why its curve is having a sigmoidal curve. Once the inhibition starts happening, it will be inhibiting further. So therefore, if there is a PFK mutation that leads to a loss of allosteric inhibition by ATP, that means what? The ATP will not be able to bind. If you have a allosteric enzyme, this is the substrate binding site and this is the ATP binding site. Now ATP site is mutated, ATP binding site. It's saying that effect of a mutation that leads to only to the loss of allosteric regulation by ATP. That means the negative regulation is lost. Positive regulation is still there, but ATP cannot bind. Okay, it will not be able to inhibit PFK. So PFK will not be inhibited. If PFK is not inhibited, means PFK is activated. It will be increasing in the activity. Okay, so out of this, as you can see, it is increase in the activity of PFK. Option 2 is the right answer. Why not decrease? Because if ATP would have been there, it would have decreased. And decrease in the amount of ADP generated by PFK. So if activity is increased, ADP will be increased, but it is saying decrease. That's why it is not correct. And ATP, PFK doesn't generate ATP. PFK utilizes ATP. That's why this is also not correct. Next question from part C. So there were total 4 questions from part B. Now moving on to the questions of part C. The first question, question ID 51. The pathway for de novo biosynthesis of purine nucleotides. Now this is the skeleton, nitrogenous base of purine nucleotides. It involves the production of IMP. This molecule is ionosine monophosphate. From here only you get the 2 other purines, that is AMP and GMP. Okay, but initially from the de novo pathway, IMP is synthesized. Now, what is the base that IMP has? This particular thing, it is hypoxanthine. Like here the base will be adenine, here the base will be guanine, but here the base is called as hypoxanthine. The structure is given here. Now, if hypoxanthine was incorporated into double-stranded DNA, which of the following option correctly represents the order of its pairing preference? We know that adenine pairs with guanine. We know that cytosine pairs with thymine. Okay, but what about hypoxanthine? What will it pair with? And in fact, what will be its primary preference? So this again is a question from nucleotides only, nucleotide biosynthesis, metabolism you can say. So efficiency of binding of hypoxanthine with bases of nucleotides, it requires a knowledge of that. Now basically hypoxanthine is keto form purine. We know adenine is 6-aminopurine. That means in the purine the 6th group is amidated. But hypoxanthine doesn't contain that amino group. As you can see here, you have a oxygen, ketocarbon. So it is a deaminated adenine basically. And cytosine on the other hand is the amino form pyrimidine. Okay, so they will combine together. This hypoxanthine, it behaves like a deaminated adenine, and cytosine, which has the amino form pyrimidine, they will make a double hydrogen bond efficiently, okay, than binding with any other bases. So cytosine has the main preference because of purine pyrimidine interaction. Okay, so it was taken from a paper only. If you see the thermodynamic stability, which one will be more stable thermodynamic wise? So first is cytosine, then adenine, and then thymine, and last will be guanine. Okay, so the correct option is option 2. Next question, question ID 52. Following statements are made regarding glycogen phosphorylase. Again a question from metabolism. So total now 3 questions are already given from metabolism. And glycogen synthase activities in relation to their phosphorylation status. Yes, glycogen phosphorylase catalyzes a reaction which is called as glycogenolysis, which is glycogen breakdown. And glycogen synthase catalyzes the making of glycogen, so that is glycogenesis. So these two are opposite processes, making of glycogen, breaking of glycogen. And these enzymes, how are they regulated? Whether glycogen is required in the cell or it has to be broken down in the cell, it is done by phosphorylations. So how does phosphorylation affect their activity? Based on that, this question is given. Which of the following combination is the correct sequence? So if you look at the glycogen synthase and glycogen phosphorylase, if glycogen synthase is phosphorylated, it is in the inactive form. But that same phosphorylation, if it is done on glycogen phosphorylase, it turns it into the active form. This is the only difference. This entire signaling is done by the regulators which are present depending on what is the glucose level, that is epinephrine, glucagon. They will help in phosphorylation. So glucagon present means that glucose level is less. And if glucose level is less, you don't want to synthesize any more glycogen. Glycogen is a polymer of glucose, the storage molecule. So glucagon signaling means glucose is less. Synthesis is not required. Hence, this enzyme has to be inactivated, the synthesis enzyme. But you need to break down glucose from already stored glycogen. So glycogen phosphorylase should be activated. But this is done by the same signaling molecule, glucagon. It activates a GPCR signaling, and with that adenylate cyclase and cyclic AMP and PKA, protein kinase A, it gets phosphorylated, but the behavior upon phosphorylation is different. Glycogen phosphorylase, it is activated when it is phosphorylated, and glycogen synthase, when it is phosphorylated, it becomes inactive. So based on that, yes, what is correct? Phosphorylation of glycogen phosphorylase makes it active, so increases its activity. This is correct. And phosphorylation of glycogen synthase inhibits its activity, so decreases its activity. So A and D, option 4 is the right answer. Next question ID 53. Dixon plot from enzyme kinetics, subunit 1F. Dixon plot is used to study the enzyme inhibition by plotting various expressions of velocity, and inhibitor concentration on the x-axis and y-axis as given below. So they have already given what is there on the x-axis and what is there on y-axis. You just have to find out, okay, like which form of the particular value is there. So if it is velocity, then what velocity? Only velocity, or 1 by v or 1 by I in x-axis or y-axis? You have to find a combination. So basically, Dixon plot, a graphical method for determination of the type of enzyme inhibition. So the inhibitor constant which we have in enzyme inhibition, Ki, it is an indication of how potent an inhibitor is. It is a concentration required to produce half of maximum inhibition. Just as how we have Km, like that we have Ki. So it is like the dissociation constant but for an enzyme inhibitor complex. How the Kd value is for enzyme substrate complex, like that Ki is for enzyme inhibitor. Now the effect of the enzymatic rate is determined at 2 or more substrate concentrations and over a range of inhibitor concentrations. So basically, if you take a plot of 1 by V against I, that is concentration of inhibitor, that is what gives you a Dixon plot. Data for each substrate concentration, they will fall in straight line. It's a straight line only. If it is competitive inhibition, then it will intersect at I is equal to minus Ki and 1 by V is equal to 1 by V max. And if it is non-competitive, then they will intersect on the abscissa, where 1 by V is equal to 0. So this is the 1 by V line, right? 1 by V is 0 over here. So they will meet over at this point. Okay. And where I is equal to concentration of I will be minus Ki, right? But here they are not meeting at 1 by V is equal to 0. So they will be meeting at where 1 by V is equal to 1 by V max. That is for competitive. This is for non-competitive. Okay. So basically, if you plot this 1 by V and inhibitor concentration, it will be giving you the intersecting lines, and that's what is the Dixon graph, Dixon plot. So what do you take in x-axis? In x-axis you take inhibitor, okay, inhibitor concentration. So column A is option 4, and in the y-axis you will take 1 by V. So column B is option 1. So generally we have a tendency to see y-axis, x-axis. So we will have a logic. So don't put 1 by V over here. If that's what you have done, then that's a silly mistake. Here correct option is option 1. Next question number 54. It is about collagen structure. So this is from protein structure, which is 1B, or you can say 1G also, subunit. Collagen has a triple helical domain structure which consists of 3 distinct alpha chains. Yes, collagen doesn't have alpha helix. It is called as alpha chain. The collagen triple helix is stabilized by isoprenyl bonds. No, it is stabilized by hydrogen bonds. There are intermolecular hydrogen bonds formed between the 3 alpha chains. So B will be wrong over here. Each alpha chain has a left-handed polyproline 2 type helix. Yes, collagen alpha chains, they are not like right-handed alpha helix. They are actually left-handed, and they have a lot of concentration of proline. So they kind of resemble the polyproline type 2 helix. Each is composed of multiple triplet sequence. The sequence which is repeated many times in the alpha chain is glycine, Y and Z, where Y can be commonly proline and Z can be usually hydroxyproline. So all are correct except B. Option 1 is the right answer. So as I mentioned that collagen, statement B is incorrect because 3 polypeptides, they are held in hydrogen bond, not by isoprenyl. And all collagens are trimeric protein. They are made from 3 polypeptide alpha chains, which are usually called as collagen alpha chain. So all or part of this 3-strand collagen can twist together into a special triple helix called as the collagen triple helix, and it can form because of an unusual abundance of 3 amino acids, glycine, proline, and also a modified form of proline, which is hydroxyproline. They make up the characteristic repeating sequence like glycine X and Y, where this X and Y can be anything, any amino acid, but are often proline and hydroxyproline in position Y. And a polyproline helix is a type of nothing but a type of protein secondary structure, which is a left-handed helix. and which occurs in protein comprising repeating proline residue. So since this has very high composition of protein residue, that's why it's the right answer. Here you can see how the three chains of collagen, they are associated with hydrogen bonds. You can see here that they are having intermolecular hydrogen bonds. So a lot of concentration of proline residues. That's why since it is left-handed, they resemble type 2 polyproline helix. And the three chains, they will be linked together to form a triple helix. Okay. Next question, question number 55. The figure below represents the denaturation and renaturation profile of a double-stranded DNA. That means before the experiment, the DNA was in double-stranded form in citrate buffer. Now the percent of DNA that remains denatured at 30 degree after cooling down from 100 degree. Here the graph has started, this temperature has started from 30 only. So what you did was you started from here 30 degree, you started heating, nothing happened, but suddenly at 80 the denaturation happened. You know that when double stranded is converted into single stranded, there is more absorbance by the nitrogenous bases. So suddenly the absorbance will increase, and that is cooperative. Once one part starts opening up, other parts also starts getting denatured. Because of that, when it reaches 100 degree, it has completely denatured. Okay, by the time it has reached 100 degree, it has completely denatured. That means none of the forms are in double stranded form. All are in single stranded form. What have you done now? You have done cooling from here. So from 100 you have again brought it down to 30. But what do you observe here? When you started the double-stranded DNA, the absorbance recorded was 1. Okay, at 30 degree centigrade. But after cooling, the absorbance came to, this is what can be considered some 1.12. Okay, so absorbance came to 1.12. The maximum renatured form, that means totally denatured form, nothing was in double stranded, it was recorded 1.40, right? So that means 100% denaturation, the absorbance noted was 1.40. Okay, and 0% denaturation, it would be 1. But after cooling, it doesn't come to the complete double stranded form. Some part of it is still in the denatured form. So this amount is still in the denatured form, right? So if we take a difference of that, we will get how much is the denatured form. So what we'll do is we'll just take the absorbance only for that. So initially when 100% of DNA in double stranded form, the absorbance is 1. According to graph, the absorbance of 100% denaturation is 1.40. If you start cooling from 100 degree to 30 degree, now if you are following the cooling graph, that is this graph if you are following, you will notice that absorbance values goes from 1.40 to 1.12 and not 1 from where it started. So this is for the DNA part of which got renatured. Okay, so that means till here it got renatured, but this difference is still denatured. Absorbance of DNA is still in denatured form, which did not renature at all. Okay, so that is 1.12 and minus 1. Minus 1 is where all of it was in double strand form. This is how much ever came down after renaturation. That means the difference is still in denatured form. So this is the amount you are supposed to find out, because the question is asking percent of DNA that remains denatured at 30 degrees centigrade after cooling down from 100 degrees C. So according to that the calculation will be 1.40 is the absorbance for when DNA is 100% denatured. Like that unitary method, if you take 1 for that 100 by 1.40, but you need to calculate for that 0.12 absorbance. How much is for that much DNA? Okay, so that will be 100 by 1.4 into 0.12, which will give you 8.9%. Okay, and 8.9% is less than 25%. That's why option 1 is the right answer over here. Okay, tricky question. If you observe the graph properly and if you note down that difference part which is still in the denatured form, then only you can get it correctly. Okay, next question. So this is again from nucleotides, the behavior of nucleotides. You can talk about subunit I, right? That is stability of DNA. and different forms of DNA. From that part it is given. Okay. Then part C, question ID 56. So there are 6 in a stretch unit 1 questions. You can very well imagine how important it is. This is a slightly doubtful question and answer also. Anyways, I'll put forward across. Hemagglutinin protein in influenza virus contains a long alpha helix with 53 residues. Which of the following correctly describes the attributes of this alpha helix? So yes, for alpha helix, you have to find out length, the turn, and the number of hydrogen bonds. Now this is what is your alpha helix looks like. This is one turn. One turn means 360 degree, where the polypeptide takes a 360 degree turn. The total length of this one turn is 5.4 Armstrong, and the number of amino acid present in one turn is 3.6 amino acids per turn. So if you see what is the distance between two residues, that will be 1.5, because total is 5.4 and how much is present? 3.6. So 1.5 is the distance between two amino acid residues in the polypeptide chain, that is in the alpha helix. So if you calculate for this protein which is having 53 amino acid residue, the length, number of amino acid residue into rise of the helix. That means how many amino acid and how much is the distance from the next. How many amino acid, how much distance. The number of amino acid is given 53 amino acid in the question and the length is 1.5. So total length will be 79.5 Armstrong. So yes, many of them somewhat it is closer, right? So we can take any of them. Number of turn calculation with 53 amino acid. So as I mentioned, a complete turn is 360 degree. Now 360 degree, in each turn there are 3.6 residues. So what is the rotation from one amino acid to other? It is 100 degree. Means if one amino acid is here, the next amino acid will be rotated 100 degree. The next amino acid will be rotated 100 degree. Okay. So to complete a full turn, 3.6 amino acid must be present. Therefore, one turn contains 3.6 amino acids. So in option, you have 53 amino acids. So simple, the 53 divided by 3.6. Somewhere around 14 turns you will get. So if you compare 14 into 3.6, yes, roughly you are getting the same number of amino acids. So yes, 14 turns you have to look. Again here in this option and this option 14 turns is correct. Okay. Now number of hydrogen bond 102 or 104. Now in alpha helix the hydrogen bond is from ith amino acid to I plus 4th amino acid. Now in this process what happens, the first 4 in a polypeptide chain, so let's say you have 53 amino acids, the first 4 amino acids, they will have only hydrogen bond with their CO groups, not with their amino group. And the last ones, that is 53, 52, 51 and 50, these 4 will have hydrogen bond with only their amino group, not their carboxyl group, because you need a 4 amino acid gap. So if this has to bond, then you would need more amino acids this side, which is not there as this is the last amino acid. So the first 4 and the last 4 are only involved in half hydrogen bond. Okay. So 4 halves means 2. Here 4 half hydrogen bonds means 2. Now in between what is left? 5 to 49. Okay. So 5 to 49 you will have 45. 45 plus 2 is 47. 47 plus 2 is 49. So ideally the answer should have been somewhere 49. That's where I felt that none of these options are matching. But yes, let's wait for the answer key challenge, what they will do, right? Now next question is actually present along with unit 13 questions. Although this is a typical enzyme kinetics question from 1F, but it was present along with unit 13. So was the next question, that is question 123, which is from the diffraction pattern. Yes, diffraction patterns are one of the techniques in unit 13, but what they have asked the question is the pattern of A DNA and B DNA, which is totally a part of unit 1. That is the forms of DNA, which means that unit 1 and unit 13 most often they give cross questions. So it's better to study them together. So what is this question 120? A student was asked to plot a graph representing enzyme kinetic data for initial velocity V0 and substrate concentration S using any of the equations below. Means they have to make some kinetic graph with V0 and S, some forms of V0 and S. Now student used an equation for which neither X nor Y had independent variables. It means that both X and Y had dependent variables. Okay, so that's where you need to have a theoretical knowledge of enzyme kinetics also. So what is a dependent variable? Velocity is a dependent variable. Substrate concentration independent. Enzyme concentration independent. But V0, Km, all these are dependent variables. That means the student must have taken in the graph some equation which involves V0. In the x-axis or y-axis, there should be some V0 involved. So which one of the following equations might the student have used? This is a very perspective type of question. You need to know the plots properly. If anyone is very well aware of their plots, this is nothing but your Line-Weaver-Burk plot, means Line-Weaver-Burk equation, which is 1 by V is equal to Km by Vmax into 1 by S plus 1 by Vmax. So this is in the form of Y is equal to Mx plus C. That means whatever you have, this is a straight line graph equation. So whatever you have in y-axis, you plot in the, means in place of y, you will take it in the y-axis. So for LB plot, you will have 1 by V0 in the y-axis. And whatever you have in x, that is 1 by S. So 1 by S will come in the x-axis. Now the student had both dependent variables. Neither x nor y had independent, means both had dependent. So 1 by S has come in the x-axis, which is becoming independent. So that means he must not have used LB plot. Okay, because here x-axis variable is independent. He had used some equation where both are dependent. Got it? So one cannot be answer. Now if anyone will know this very popular one, Michaelis-Menten equation, V0 is equal to Vmax into S divided by Km plus S. Now everyone knows Michaelis-Menten, we use velocity in the y-axis and substrate concentration in the x-axis. Again, S is a independent variable, so he hasn't used any independent variables. That means even Michaelis-Menten cannot be the answer. Then the third kind of equation that they have given is Haynes-Wolff equation. That is S by V, 1 by Vmax into S plus Km by Vmax. Now if you see here, same y is equal to mx plus c. If you apply, S by V is y. So in y-axis you have S by V, and this x substrate will be in the x-axis. So again you can see that substrate, which is a independent variable, it has come in the x-axis. That means you would not have used Haynes-Wolff plot either. So where is the Haynes-Wolff plot? This is the Haynes-Wolff plot. So this one also he must not have used. So the only one which is left is this one. Now this one is actually a derivation of another plot which is called as Edie Hofsty plot. Okay, this is Edie Hofsty plot. This is the equation for it. This is a slight modification of the equation. So they have taken S here. If you just cross multiply, means if you just bring the S over to the other side, so divide by S, so that way you will get the equation. But whatever it is from this equation, if you plot the graph, then you get V0 in the y-axis in the form of y, and in the form of x you get V0 by S. So this is what is, both are having dependent variables. As you can see, both are having V0 as part of the axis coordinate. So that means the student must have used this one. It doesn't contain any independent variable. Okay, so correct answer over here will be Edie Hofsty plot. Again, a pretty tricky question. You not only should know about the Line-Weaver and Michaelis-Menten, but also these knowledge is required. And another question along with unit 13 questions, this was diffraction pattern of A DNA and B DNA. And they have given some forms actually. They have not mentioned which one. Form 1 and Form 2. So you have to identify which is what. Okay, and not only that, you also have to identify which form has what bases, what number of bases. Now if you look at, if you know about the diffraction pattern, yes, Franklin Rosalind and Raymond Gosling discovered the extra diffraction pattern, And according to their experiment, this A and B, they were given this diffraction patterns. So you will see that the 8th layer line in A occurs about the 10th layer line of B. So these two were different forms. It means that there is a 20% increase in the DNA helix period. If you would know that what is DNA, bDNA, aDNA is actually a compressed form of it. Okay. So yes, here we can see there is a increase in the helix period in bDNA. Then aDNA pattern shows crystalline spots on the first few lines, first few layer lines. Okay. But B shows broad streak. So you can see here these are some crystalline spots, but here is a broad streak. A further in the A1, significant difference is there between the two patterns near the center of the X shape. So you can see here in the first three layer lines in B, so that is not present in A. That cross one is not present in A. Now the X shape is produced by regularly repeated part of the molecule, namely by its helical sugar phosphate backbone. So X shape appears clearly in pattern B, which reflects the scattering by an individual angularly averaged molecule, whereas it cannot be discerned in pattern A because the pattern is resolved in individual crystalline spots near the center. And then B also shows a diamond pattern, which not only means it is helix, but it also reveals the three fundamental dimensions of the phosphate helix, its pitch and all that, right? So if we compare, after completing the qualitative description of aDNA pattern, it was found that in aDNA, the number of nucleotides per helical period is around 11 bases per strand instead of 10, which is in bDNA. So bDNA base pairs per turn of helix. In one turn, how many bases are there? For bDNA, it is around 10.4, and for aDNA, it is around 11. Since it is compressed, more nucleotides will be accommodated in one turn. So according to that, yes. So diffraction pattern of aDNA is shown in form 1. That's right. No. So the diffraction pattern of bDNA is shown in form 1. So this is what is the cross part, which is bDNA, and it has 10 bases per turn. So D is definitely correct. And aDNA is form, this you can see crystalline lines. aDNA is form 2 with 11.6 bases per turn. So C and D, option 1 is the right answer. So again, this is from nucleotides only. Pretty scattered type of questions are there. Ramachandran plot did not have any questions. Other hydrogen bonding, hydrophobic interaction, pH, buffer, thermodynamics, even lipids did not see any questions. But again, these proteins were more expected. Nothing with that, only collagen structure was there. So that's all for the discussion. Thank you, everyone.